All right, everybody. Good afternoon and welcome. My name is Richard Mitchell. I'm the director of the Kids Cancer Centre at Sydney Children's Hospital, and I welcome you to our Advanced Therapeutics Webinar series, where we focus on the rapidly evolving space that is advanced therapeutics, as it prevents innovative and potentially transformational treatments for children now and in future years. Before we get started, I'd like to begin by acknowledging the traditional owners of the various lands on which we're all meeting today. Sydney Children's Hospitals Network acknowledge that our facilities are built on the lands of the Bidjigal, the Camarail, the Burramatagal, the Kabarabal, the Wanagal, and the Wadi Wadi. Our services extend across the lands now known as New South Wales. Aboriginal people believe these lands hold the ancestral spirits of those who have come and gone before them. We acknowledge that this was and always will be Aboriginal land. Thank you, Jing. I'll get you to stop screen sharing. Today, we're going to be hearing from Associate Professor Emily Blyth and Professor Geraldine O'Neill. Associate Professor Emily Blyth is the lead of the Immune Effector Cell Service at Westmead Hospital, and Professor Geraldine O'Neill is the head of the Children's Cancer Research Unit at Kids Research at the network. Their presentations will explore cell therapies at the point of care for children and young people. And then following these two fabulous presentations, we'll move into a Q&A session to foster some discussion. So please submit any questions to the webinar via the Q&A tab in the Zoom controls. And if you see a question submitted that you'd like to have answered, you can vote the question by clicking the thumbs up beside it. I now have great pleasure in welcoming my friend and colleague, Emily Blyth, to begin her presentation.
Good afternoon, everyone. Thanks so much, Richard. Thumbs up if you can hear me. Yep. Let me just share my slides now. Sorry. There we go. You should be able to see those slides there with that intro slide.
Not yet, Em.
Let me see. Okay, let me see if I can fix that. It worked before.
Yeah.
Okay, how about now?
You just need to just switch displays.
Okay. Sorry, I'll just do that again. It was seamless the previous time. Here we go. Okay, we good?
Perfect.
Okay, great, thanks. So I really appreciate the opportunity to talk to you all today. This is such a fast-moving field, and it's really important for us to keep alert to all the things that are happening. So this is a bit of a Westmead showcase within our very dynamic New South Wales cell and gene therapy environment. So my job is, I've got several different roles. The one that takes the vast majority of my time is to run the CAR T cell service at Westmead, and with a future focus, we call it the Immune Effector Cell Service because we know that CAR T cells are not the only immune effector cells that will be available for patients as we go forward. So I have a number of other roles, including I'm also a University of Sydney research academic and the current regional vice president for the International Society of Cell and Gene Therapy, and I represent the ANZ region in that group. All right, so Geraldine and I both are going to talk about chimeric antigen receptor T cells today, which is one of the quite large group of advanced therapies. And when I say large, I mean mostly in the sense of the quantum of development in terms of the number of products in the pipeline. And it was one of the first to reach routine use. So this slide here has a little, I guess, a schematic illustration of what chimeric antigen receptor cells are. Effectively, they are immune cells that have been redirected in various ways to target something other than their original immune effector cell activity. So in this case, T cells that normally fight infections or natural, the tumors as they develop in our bodies that have been redirected to a particular target. And the ones in dark blue or dark greeny blue are the ones that we use routinely in Australia. So we have 3 products, CAR T products that are available commercially for B-cell malignancies, leukemia, and lymphoma in children and adults. We are literally about to get CAR T cells for myeloma. It's been a long time in the pipeline, and I believe those contracts are going to be signed anytime in the next few days. But there are lots and lots in the pipeline for other hematological cancers, a whole bunch of other cancers, autoimmune diseases that are mediated particularly by B cell and plasma cell elements, CAR T cells for HIV, and then, of course, combination cell and gene therapies for genetic diseases and many other things. So this slide, which shows active cell therapies, this is just data I could find from 2024, shows the increase in the number of total trials for cell therapy. And this is globally. In 2024 cell therapy trials, there was 500 or 600. And this is a slide meant to dazzle you for the different products in development for gene therapy. So none of us can read any of that stuff. But the point being that there's a huge amount of activity in this space. And if even a fraction of these therapies demonstrate efficacy, safety enough to reach routine care, it's really going to change things for us all. So looking a little bit more detail about, these are all the cell-based gene-modified therapies that are available globally. And this was current to the end of last year. There's since been some approvals, so it's not even current at the moment. So what you'll see here is that in Australia, we have approval and funding for 3 different products, Kymriah, Yescarta, and Tecartus. Cilta-cel being the one that I mentioned, the myeloma one, that's a funding agreement's about to be signed. But in Europe and the US, a lot more are available. So what's unique about Australia, that means we can offer our patients less options in this category of high cost, low volume advanced therapeutics. So let's quickly go through what happens in Australia. We have a health system that provides close to free care to our community, citizens and permanent residents via Medicare and the state government funded hospital system. And everything that is provided and funded for our community goes through processes of vetting. First of all, TGA approval is given, focusing mostly on safety and efficacy. So a product needs to be presented to the TGA, and there needs to be data to support its use in terms of those 2 features, particularly efficacy and safety. And then either the Pharmaceutical Benefits Advisory Committee or the Medical Services Advisory Committee has a look at cost effectiveness and whether it is reasonable for our health service to pay for it. Is the cost worth the outcome? So CAR T cells have gone down the medical services funding route because there's so much clinical care that's associated. You don't just get them off the shelf and give them to patients without a whole bunch of other health service resources being utilised. So medical services then assesses it for whether we should fund it. And then there's a process of of Commonwealth, state, and health service agreements, and eventually then it gets into clinical care. So that process can be lengthy, and this forms part of the reason why we in Australia can only offer a small proportion of the ones that are available globally in the market. So the way that this has been rolled out in Australia is via a network of CAR T centres, and you'll see that I've got very inaccurate placement of each of these locations. So we have 10 adult sites, 5 paediatric sites in New South Wales, which looks like I've placed them approximately in Coffs Harbour, and all the way out west to, I don't know, that looks like it might be Tamworth, but we have the Sydney Children's Hospital network hospitals here in light green, and Royal Prince Alfred and Westmead. We hope that we will be adding another 2 adult sites fairly soon. So this is, we have coordinated nationwide eligibility assessment. There's a combination of Commonwealth and state funding. And in New South Wales, there is also a very functional and world-class network of blood transplant and cell therapy. So you guys are part of that network, which is run by the ACI, so the BTCT network, and that is actually quite unique around the world, in the sense that it has centralized quality management. It's got consensus clinical guidelines that are available to all the hospitals in the network. It's got clinical and laboratory working groups that ensure that all of the standard operating procedures and guidelines are up to date. It runs education forums and it provides patient resources. So it's a really fantastic way of ensuring high quality care and uniformly high quality care. So it includes 13 clinical sites and apheresis units and 7 stem cell processing labs. So some of the labs support several hospitals, including at Westmead. We support 4 hospitals with our stem cell transplant laboratory. And a number of those services are FACT accredited. So the 2 Sydney Children's Hospital Network hospitals and Westmead Hospital are the 3 FACT accredited. That's a foundation for the accreditation of cellular therapy, and that's an extremely rigorous, internationally recognized accreditation for high quality care. So that's what we do there. And at Westmead, that's New South Wales wide. And then at Westmead, we have a number of different activities. So Geraldine's going to talk about the preclinical research and a little bit about our translational laboratory, the Human Applications Laboratory, aka Sydney Cell and Gene Therapy. We have the Blood Transplant and Cell Therapies Laboratory that supports the Children's Hospital, Westmead Hospital, Wollongong Hospital, and Nepean. And then we also have at Westmead Adults Hospital and the Children's Hospital both have FACT accredited clinical programs. So when this was established in 2020, we needed to create a whole support network for the patients that included doctors, nurses, critical care support in ED and intensive care. We have a pharmacist, neurologist. We have scientific staff in the laboratory. We obviously have operations. And then I haven't put it here, but we also have a very strong social work network because our patients come from all over New South Wales and sometimes other states to have treatment with us. So that's what's required to treat just one patient. But with all of that established, then we have the capacity to do lots of different things. So I'm going to just show you one efficacy slide here. So this type of arrangement that we have at Westmead is duplicated all around Australia in the CAR-T sites. And our national data has recently been presented and published in abstract form at the American Society of Haematology meeting. So this is just one snapshot. CAR-T cells for B-cell malignancies are potentially curative in patients who have refractory and relapsed disease. So these are patients who failed other therapies, and the survival usually is very poor, in the likelihood of sort of 10% in a sort of 12 to 18-month period. So what we see here in this Kaplan-Meier curve is patients who are treated with one of the products available to us, axicabtagene or Yescarta, have got a 2-year survival of just under 50%, which is a vast improvement previously. I will note a vast improvement in what they would achieve without this treatment. I will note, though, that there is still a long way to go because this part of the graph are all the people for whom it was ineffective or effective temporarily, and then the patients then had relapse of their disease or a bad outcome from toxicity. This particular product is one that has got significant toxicities, and the types of things that we see with these therapeutics is they are active immune cells, they are live cell products. They go into the patient, they seek out the tumor and they replicate, proliferate and release cytokines that then induce these toxicities. So cytokine release syndrome, CRS, or immune effector cell associated neurotoxicity syndrome, or ICANS, are the 2 big toxicities that we see. And in grades of severity, with grade 5 being fatal, you can see that if we add all these up, only 9% of patients don't get any CRS, and 55% of patients get some form of ICANS. So those that have grade 0 or none are actually in the minority. And so managing the patients through what is usually temporary, it's treatable, but it requires a lot of resources. So what that tells us is that there is so much room to improve, even with the most successful CAR T cell products. We would like safer, we would like to maintain efficacy or improve efficacy, but have the patient experience being, I guess, an easier ride and, not to have to put them through the toxicities that occur at the current time. Having said that, though, the more severe toxicities are infrequent, and even those are largely reversible. All right, so where are we with the current arrangement? So currently, we're in Australia, we're far away, wonderfully, from a lot of the things happening in the world, which sometimes is a blessing. But in this situation, the products are collected from the patient, the apheresis of lymphocyte material, it's shipped to the US. The gene modification occurs there, and then it's shipped back. And that takes about 3 weeks minimum. So median time is around 20, 21 days. So the turnaround time is an issue, and patients who have rapidly progressive disease sometimes don't make it to their treatment. And I will point out here, this is 422 patients Australia-wide who were enrolled and had their lymphocytes collected. Of those, 395 received their treatment. So there are some patients who, for various reasons, one of which is turnaround time, don't end up receiving their treatment. One of the big issues is the cost to manufacture, which is not the same as the cost to purchase. So that's something that we are really going to need to address as a community as, all of those therapies under development hit the market. The time in Australia from approval to funding to patient access is slow compared to other places, and that gives our community and our patients less choice. We also have a situation where, because of the process of assessment and price negotiations that occur, we have fewer products marketed, not because we wouldn't want to pay for them, but because it's a business decision of the company that markets them not to come to Australia. As larger numbers of applications come our way, the cost that we are currently paying, the advertised price is, advertised price, which is the public price. We don't actually know how much the government is paying, but it's in the order of magnitude of $300,000 to $500,000 per dose. Dose per patient, $300,000 to $500,000 just to purchase the product. So as larger number of applications become available, nobody can afford that. We can't afford that. And nor can any other advanced economy, let alone low and middle income countries. So the issue of healthcare sustainability then becomes a question. So this is a beautiful figure. I really love it. I'm sorry I forgot to put the reference down the bottom, but this is a health economic study looking at the cost in US dollars of that product I've just mentioned, Axicaptogene. So this is where it is currently sitting in terms of the cost effectiveness or cost per quality of adjusted life year saved. We would really like to move it right down here into the green or the blue in terms of cost effectiveness. So that is what we are working very, very hard to do. So one of the alternative ways of doing this is the way that we've done stem cell transplant, the way that we've done blood banking. So to do things within the healthcare system. So is it possible to successfully make a CAR T cell in an academic or healthcare setting for routine use? And there are a number of places around the world who have been looking at this for quite some time. So I'll just provide one example. This is a publication that's actually only relatively new, just from the Lancet 2026, but it's one of the studies in patients with acute lymphoblastic leukemia using a product that received market approval in Spain in 2021, which is this one called RE001, which is a bit of a mouthful. So they've now given it a name, Captogene. So that one was developed within the hospital clinic of Barcelona, and it's manufactured in Spain by a network of academic manufacturing facilities. It is reimbursed in the same way as other therapies. The government pays per dose for the whole process of collection, manufacture, and delivery, and it shows efficacy and favorable safety profile in comparison to the commercial ones. So, because this is a really big issue for everyone around the world, there's been quite a lot of interest in the idea of, is this way of delivering therapy as effective, as safe, and is it cost effective? And the Netherlands government is actually funding a study comparing RE0001 with the commercial CAR Ts available in their country. And that is a non-inferiority study aiming to see whether it's feasible to deliver the care in this way. So we think this is fantastic, a fantastic model that potentially is applicable to Australia, where we have, the universal healthcare system that we have. So as part of that, I'm going to just give you a little bit of background to what Sydney Cell and Gene Therapy does. So that's the manufacturing facility at Westmead. So we're a GMP manufacturing facility that has been a research facility for some years. We've got 15 years experience in the entity name, but in actual fact, the first patient treated with a product manufactured at the Westmead manufacturing facility was in 2003. So it's now 23 years ago. We've supported 15 phase 1 clinical trials and manufactured more than 400 products, treated 350 patients on 15 clinical trials, and also provided quite a lot of therapy under compassionate access use. We've got a platform agnostic system here, including a multiple different manufacturing platforms. So Cytiva, Rotea, the CliniMax Prodigy, which a lot of people will know about, that's used by a lot of those things. And as part of our network, we are then also part of the Collaborative Centres Program for Advanced Therapies, which is a collaborative network around Australia that's looking at delivering not just for research, but for routine clinical care, this type of therapy for Australian patients. We have quite a lot of support and partnerships, to see if this will work. And I'll just highlight this company here, Caring Cross, which is a not-for-profit company that aims to bring cell and gene therapies to low and middle income countries, as well as it's sort of external to a pharma model. It's a not-for-profit model, and we're working with them to bring some products to the market. Now, in the interest of time, I might skip past this slide in detail, but essentially there are different ways to do this type of therapy. The pharma model is fantastic in some settings, particularly when there's a large market and common diseases. But for rare diseases, pediatric diseases, and when there's a large number, we need alternative models. So the Centre for the Collaborative Centres program is looking at several different projects. These two here partnering with Hospital Clinic of Barcelona and Caring Cross to roll out myeloma and B-cell directed therapies. Those are actually supported by, so far, funding committed by Western Australia and New South Wales governments. We also have grant funding for other projects. And just with a paediatric focus before I finish, I'd like to just shout out to this particular advanced therapy that Caring Cross has now licensed from Boston Children's Hospital, which is an advanced therapy that combines gene therapy with cell therapy, where patients with hemoglobin disorders have a bone marrow transplant with stem cells, blood stem cells that are gene modified to repair the deficit. And these are well-proven technologies with commercial options available, none of which has ever been even considered to be marketed here in Australia. Extremely expensive, $3 million per patient. And they have, despite evidence of efficacy, failed to be commercially successful basically because of that cost. And potentially these patients could receive therapy using a different model via a not-for-profit healthcare-based delivery. So as I finished, I think the value of investment in this type of therapy for our community is quite significant. It has a public good outcome. It can potentially provide a lower cost of goods, an alternative path to market, which will in turn increase the choice in our market and for our patients. And it will have the effect also of providing workforce development benefits and give us the sovereign capability to make choices that are important for our community. So are there any questions? Or we're having questions afterwards. So I was told that 10 times. But what I'll do now is I will introduce Geraldine O'Neill. Thanks so much, Geraldine.
Thanks very much, Emily. I'll just share my slides as well. Okay, great. Well, thanks. I'm so grateful to Emily to give such a fantastic introduction to the idea of place of care manufacture and a really nice overview of where the field is at. And what I'm going to do is tell you a little bit about a phase 1 clinical trial that we're running in our unit using place of care manufactured CAR T cells. So the work is being carried out in the Children's Cancer Research Unit at the Children's Hospital at Westmead. And most of the work most of the heavy lifting is being done by our Advanced Cellular Therapeutics team, headed by Dr. Kavi Garishankar. So just to briefly recap, I know Emily's done a great job in introducing CAR T cells to us, but the process is to isolate or take blood from the patients, isolate their T cells, and importantly expose them to a virus that encodes the CAR, or the chimeric antigen receptor, that will recognize the molecule on the cancer cell. We then have these manufactured CAR T cells that should be expressing the recognition domain on their surface. Those CAR T cells are then expanded and then they're reintroduced into the patient, where they hopefully seek out the cancer cell that expresses the target on the surface of the cancer cell, and that should then result in the killing of the cancer cell via the normal action of the T cell. So it's a kind of a living medicine, we like to think. So one of the things to highlight is that children's cancer is really considered to be a rare disease, and this is in terms of numbers. So lots more adults have cancer than do children. And so this, I suppose, raises some issues around the developing of therapies for children with cancer. And in particular, in the CAR T cell world, the commercially developed CAR T cells really require very large target patient populations. And Emily kind of touched on this, in order for the commercial interest to be able to recoup their upfront investment. So there's often not a lot of appetite for developing these kind of very specialized therapies for rarer indications. And so children really require specialized therapies that are able to serve these rare patient populations. And that's where our work that I'm going to tell you about today came in. But I first just want to give you a little glimpse into the actions of CAR T cells, because I think seeing is believing. So the panel over here that I'm showing you is basically in the control well, just a lump of cancer cells, and underneath is a lump of cancer cells the same size that has been exposed to CAR T cells. And the colour that you can see indicates to us how many cells are there. So the brighter the colour, the redder it is, and the larger the spot, that tells you that there's a lot more cancer cells. So you can see that when we've added the CAR T cells to this lump of cancer cells, we've really significantly killed those tumour cells. And over here on your right, I have a video of some CAR T cells killing some tumour cells. So tumour cells shown in purple, CAR T cells shown here in green, and the tumour cells will turn pink after they had interacted with the CAR T cells, and that has induced them to undergo death. So I just ask you to focus on this area in the red here as I start the video. So you'll see that green CAR T cell touching the purple cancer cells, and then subsequently they turn pink as they've been killed by the CAR T cell. So this is what should be going on in our body if we've got an effective treatment, that the CAR T cell that we've introduced will kill the patient's own cancer cells. Oops, sorry, went one too far. So this work really came about a few years ago when Dr. Jeff McCowich, who is a senior consultant in our Cancer Centre for Children at the Children's Hospital at Westmead, came with a bit of a problem for the team. So Jeff, as part of his practice, he treats children with sarcomas, so soft tissue and bone cancers. And so just to give you a little bit of background, this is a condition that commonly is most common, in teenage years. The majority of the cancers are localized, but there are a number of patients who will go on to have metastatic forms of these cancers. The treatments that are currently used are chemotherapy and surgery. So survival's around 70% if the patient has non-metastatic forms of sarcoma. However, once there's a progression to metastatic forms, this prognosis becomes poor. Similarly, for patients who experience recurrence after a primary therapy, the prognosis is poor. Another thing that Jeff really emphasises to us is how toxic the treatments are that these patients are exposed to. So the sort of side effects that result from the currently used therapies can include things like infertility, secondary malignancies, and cardiac damage. So I think it was 2017, Jeff came back from a conference overseas. We were very excited at the thought that perhaps CAR T cell therapy might be something that could be applied for his patients in the sarcoma patients that he sees, for which he has no other alternative therapy choice. So he's particularly interested in this target, FA2, and he was interested because he had met Stephen Gottschalk from St. Jude's, who had developed a vector. If you remember, I spoke about the CAR T cell is created by introducing a virus that encodes the CAR into the T cell. And so Stephen Gottschalk had produced an FA2 targeting CAR vector and was willing to share it with Jeff. We knew that FA2 is highly expressed on a range of different cancer cells, and we confirmed that there was a significant expression of FA2 in both Ewing's and osteosarcoma patients in our clinic. So on the strength of this, the team went ahead, and these three here did all the heavy lifting for this work, where they undertook a number of years of preclinical investigation of the efficacy of the CAR T cells. So I'm not going to show you all of the work that they did. I just wanted to highlight, I suppose, what is the most significant result in terms of being able to move to the clinical trial is where they took two different models, an osteosarcoma and a Ewing sarcoma model in mice, and they exposed these mice to CAR T cells. And what I just want to emphasize to you here is that post-treatment with CAR T cells, the tumours were undetectable in both models. And they also saw that there was a significant increase in the lifespan of the mice that are exposed to the CAR T cells. They also did some analysis of the potential for the CAR T cells to inhibit metastasis, and were able to show that there was significantly fewer lung tumours forming in these mice, suggesting that the CAR T cells were also effective against metastatic forms of the disease. So we were pretty excited about this data, and on the strength of this information, we moved to undertaking a clinical trial. So the clinical trial, sorry, I just have to hide my controls first. I'll just move that, yep. We spent a lot of time, of course, writing grants to get money to be able to undertake this clinical trial. So it's not a cheap thing to do, and so we sourced money from the national government in the form of a Medical Research Future Fund. We've been very generously supported by Luminous Alliance based here in New South Wales. We've also been supported by the Sydney Children's Hospital Foundation, who've raised money from donors for us. Also had funding from the Kids Cancer Project, and we've also been generously supported by the Tie-Dye Project. So all of these, this source of funding we put together into a big pool to be able to launch our clinical trial. So the trial is sponsored by the Sydney Children's Hospital Network. We'll be treating children with osteosarcoma or Ewing sarcoma who have either relapsed or are refractory after the standard treatment. The purpose of this trial is to undertake the first in-human FA2 CAR T-cell trial for sarcoma. And of course, the primary objective of this phase 1 trial is safety. Can we safely undertake the work that we plan? But our secondary objectives, which I think are equally as important, is to assess our ability to locally manufacture the CAR T cells for administration. And this is really key to support future applications of this in-place manufacture of the CAR T cells. We also, of course, will be assessing functional capacity of the CAR T cells in the patient. It's a single centre study design. We'll be looking at feasibility and safety. We're underpowered to do efficacy, so that's simply an exploratory objective. We're only recruiting a small number of patients. This is a big undertaking, and we're really excited that we, fingers crossed, are going to be opening our trial in Q3 of this year. Really can't wait for this to open and have the first trial patient enrolled. So a couple of key features of our trial. Firstly is this idea of place of care FA2 CAR T-cell manufacture. So we'll be doing that with the assistance of the Sydney Cell and Gene Therapy team at Westmead. We wouldn't be able to do the trial without this assistance. And the other sort of key feature of our trial is that we're using also a vector that was locally manufactured, and it was synthesized by the Gene Therapy Research Unit at Children's Hospital at Westmead, headed by Professor Ian Alexander. And so that's quite unique in terms of normally for this kind of trial, you would have to purchase your virus at great expense from an international company. And the Gene Therapy Research Unit have now sort of transitioned into the local viral vector manufacturing facility. So hopefully in the future, it'll be possible to buy these viruses for trials and treatments locally. So again, addressing this kind of sovereign manufacturing capability. So because we're doing the local manufacture of the cell therapy, there's a lot of considerations around the regulatory processes and that we have to undertake. So the CAR T cells are classified as a class 4 biologic, and that means that all of the processes involved in manufacturing these cells is highly regulated. So under GMP or GMP-like manufacturing regulations, we have to have, of course, quality controls and quality assurance. All must be done in clean rooms, and the protocols that we use to both make the vector and introduce it into the patient cells must be under very strict, standard operating procedures. And so all of this, of course, takes a lot of time to do, and we've spent a lot of time building up the bolus of evidence that's required to be able to transition to clinical trial. And it takes a team to do this, a big team. These are just some of the personnel. I guess these are the people who are directly employed to undertake the clinical trial. So the clinical trial is led by Jeff McCowich, so an oncologist in our cancer centre, as I mentioned, and co-led by Dr. Kavi Garishankar. So Kavi really oversees this scientific team who are doing the work, and we have a number of clinical appointments associated with the trial as well. So all of this is just to say that undertaking this work takes a large team, a large dedicated team, including the quality manager and quality control scientist activity that is undertaken by the Sydney Cell and Gene Therapy Unit. And so I just want to highlight down the bottom right-hand corner, Amanda Tan and Catherine Dodd, who are our manufacturing scientists. And so they're doing the very important work of transitioning the protocols and the work that we're doing from our research laboratories into the clean room that Emily mentioned that's required for the trial to begin. And so the next slide I'm going to show you is a slide that I borrowed from Amanda, the sort of lead manufacturing scientist. And Amanda's, as she's indicated here, well, I've got all my research methods set. You know, I should just be able to transfer that into a manufacturing SOP directly. It turns out that's not the case, and that's because there's a number of steps that need to be quality controlled and validated in this transition, including things like the scale up of the T-cell isolation. There's also the manner in which you activate the CAR T-cells is different in the clean room facility. There's the issue of scaling up the vector production, and then how that in the multiplicity of infection of the vector. You know, we need to achieve a certain level of CAR T transduction in order to have a viable product. There's also then the upscaling of the CAR T cells themselves. Every time you go from the sort of the small research to the large therapeutic application, it comes with complications in terms of working out the methods in a very organized fashion. There's also then what happens to the product while waiting to be delivered to the patient. So while the cells might have been made, they have to be sent off for testing to make sure that they're sterile, that it's a safe product to deliver to the patient. And so that can require freezing down of the cells in between, and all of that must again be done under very strict regulations and guidelines. So finally then is the infusion into the patient, and the manner with which that's done will be determined by the clinical protocol. So as Amanda's highlighted here, each step requires a specific solution, and that solution is very much determined by the clinical and regulatory requirements that surround this manufacturing process. So Amanda's slide here, where to start. This is a slide from a little while ago. I'm happy to say that a lot of this work has been completed now. And this workstream slide is really just to show you the very complex setup that is required in order to proceed to the clinical trial. But again, happily, much of this work has been done now, and we're really down to this stage where we're preparing our dossier of information for the Therapeutic Goods Administration. So we have to prepare a clinical trials application, and that should be submitted shortly. And once that is in and approved, then we will be at the point of first patient enrollment, and there will be much cheering in the lab once we get to that point. So I just wanted to then highlight how this work that we're doing wouldn't be possible. It's very dependent on the Westmead situation, our geographical localization. So there has been much resourcing, infrastructure and expertise developed in the local Westmead precinct that really facilitates us in doing this clinical trial. I've just listed here some of the sort of major achievements in the Westmead precinct, but actually I just want to highlight that the Westmead area is really known for being leaders in stem cell transplant for both adults and children. And it's really on the background of that environment that there's been this substantial investment in the viral vector manufacturing facility, and also in the Sydney Cell and Gene Therapy facility. So this, you can see how this ecosystem has really been built up to really allow us to undertake this place of care manufacture for children with cancer. So I just wanted to finish up by showing you the key points that I think are arising from our trial. And actually, this is part of a larger plan of driving CARs and other immune cell therapies into the clinic for treatment of children with cancer. We're really trying to develop a pipeline of local innovation and manufacture, so we've got that whole pathway for patient application. So our trial, as I mentioned, is the first in human FA2 CAR T cell clinical trial in sarcoma. It's been endorsed by our consumer advisory group. We take the opinion of our consumers very seriously. We try to ensure that all the work that we're doing is patient-centered. Our trial is really distinguished by the fact that we're using an on-site manufactured lentivirus, and we're also doing place of care CAR T-cell manufacture. And again, one of the key outcomes to come from the trial, beyond hopefully curing some children with sarcoma, is that we are helping to build the capacity for this place of care manufacture at Westmead. That's a really critical need for children with cancer, given their rare disease status. So, of course, I'd like to finish by acknowledging many, many people who've been involved in getting this clinical trial together. My own unit, the Children's Cancer Research Unit, and the Advanced Cellular Therapeutics team, of course, but many of the unit contribute to this work, including our consumer advisory group. But we also interact broadly with a number of departments in the Children's Hospital and wouldn't be able to do this work without them. So the bone marrow transplant unit, pathology, our research office team. And we're also very grateful to the facilities that are located on the Westmead precinct. As I've mentioned, the viral vector facility and the Sydney Cell and Gene, and particularly highlight Leighton Clancy, who we work with very closely. We're also very grateful to our colleagues at the Westmead Adults Hospital, who are generous in terms of sharing their knowledge and expertise with us. We acknowledge Tessa Gargett, who is the scientist responsible for the Levi's Catch trial, and she's been very generous again in giving us advice about getting our trial up and running. And finally, to Stephen Gottschalk, who shared the vector with us that enabled us to do to make the FA2 CAR T cells. So thanks very much for your time. I'll stop sharing, and I am going to hand over to Richard Mitchell at that point.
Thank you very much, Geraldine, and thank you for a fabulous talk and looking at an exciting new therapy that's going to be hopefully soon available, and also to Emily for her wonderful talk as well, giving us a look at the landscape locally as well as a little bit wider. So my next introduction is someone hopefully you all know. So Michelle Lorentzos is the medical lead for the Kids Advanced Therapeutics program at the network, and she's in charge of facilitating Q&A. So I'll leave that to Michelle. Thank you.
Thank you so much, Richard, and excellent. We can see Emily again as well. Yes, so thank you so much for such elegant depictions of an area of technology that is becoming such an exemplar for us in advanced therapeutics, both in terms of understanding the science of precision medicine, but also the translational challenges that you outlined. There's lots of interest, and we had over 115 participants at one point, with most of them still on, which is wonderful. So can I start by asking one of the questions that's in the chat, and a reminder to participants, you're welcome to both write questions as well as upvote them in the Q&A. The first question is for Emily, and it's asking about the manufacturing costs and turnaround times that you mentioned as being major challenges. And do you think the field is open to adopting novel ex vivo or in fact any other enabling technologies that may help streamline manufacturing coordination, particularly in point of care settings?
100% emphatic yes to that. The costs that we talk about for the purchase price of the commercial ones, and Geraldine, she also mentioned that the things that add into those costs are the scale and the market size, and not just the cost to actually produce the thing. So it's about recouping of research investment. The pharmaceutical model really relies on scale up. For example, you develop something that's really complicated and difficult to develop, but then once you finally manufacture, you can scale up to millions of doses potentially at a relatively low cost. But this just never will get there. So the cost to manufacture has some major kind of components. So there's all the technology, there's reagents, the viral vector, which is the gene modification mechanism. There's all different kinds of technologies there, lots of different things in the pipeline. Those ones can be very expensive. So we go for we estimate between sort of $40,000 and $60,000 per patient for just that reagent. With the Caring Cross model, they have a price that's much lower than that, in the sort of teens, I think, for the vector that they are supplying, and they also have worked up a platform of manufacturing that's extremely efficient. And so the products there will have a overall reagent cost of around $50,000. The Barcelona ones, which includes apheresis, is about 100,000 euros for the whole thing. So that's the cost to purchase that. And that includes all the staffing and the infrastructure and all that sort of stuff. So we think that we can get an order of magnitude lower cost to produce these products if we go with a different model. The question about are we willing to, or is the field willing to adopt these new technologies? One of the ways in which democratization or decentralization of manufacture will be possible is if there are automated systems with fewer human variables. So the Prodigy system is an example, but they're absolutely not the only one. That's made by a company called Miltenyi, but Lorentzos, Thermo Fisher, Cytiva, all big companies that each have a platform that will allow automated manufacture. So the way I see it will be like you'll have your CAR-T kind of Xbox or PlayStation, and you stick in your cartridge, your vector goes in, your reagents go in, and what we buy is the pieces, whereas we have an Xbox at home, or we have a PlayStation at home or in our lab. So that's how I see it working for these rare diseases, potentially ones that don't want to come to Australia, these special indications we might want to have as a community. Hope that answered that question.
No, absolutely. Thank you. A complex question that you've answered very succinctly, actually. So I might go to Geraldine with a question from Eden Robertson. And this question is really regarding the, increasing complexity of treatment options that we have for patients and then the impact of that on communication and consent. So, I mean, I guess chemotherapies in general are quite complex, but this is a whole other level in terms of informing patients about the impact of cell-based therapies, even about the development of these therapies. And so how does this affect your approach to enrollment and consent? And is it something that you've thought about that requires a different approach to the traditional models?
Yeah, thanks, Eden. It is a really interesting question. I feel like it's a slight Dorothy Dixer for the CAT program because one of the things that the CAT program has done is develop some resources around what is CAR T cell therapy, and that's available on the website. So I think that's a really good starting point. And then the other thing, I guess, of course, we have developed our patient information sheets and with our consumer group. So to help us to try and make sure that we are explaining the bigger point, I think, about how much to explain about. I think that's a really interesting point, Eden, and I think it's something that really needs to be thought about. What are the long-term implications of using these kind of therapies? And it does need probably greater education, for want of a better word, of the patients who are going to be receiving these therapies. So yeah, really great question.
Excellent. And you both may be able to answer this, but how long do the E2CAR persist in the preclinical models that you've developed?
Oh, sorry.
That's one for Geraldine for sure.
Yeah. Yeah. Sorry. Yeah. So we don't know because, of course, we have to kill the mice at a particular time, right? So we're not able to just continue out to see how long. So in terms of in our preclinical models, we don't have particular evidence about the persistence of the CAR cells. That's something that, of course, will come out of our clinical trial. That's one of the things we'll be looking at. How well do the cells persist? We'll be looking at all those markers of exhaustion and all that kind of thing. So we'll get an absolute plethora of data out of the clinical trial.
Fantastic. And we have a question here from Dr. Jeanette Lee. I might take this one to Emily initially. So both speakers have spoken quite transparently about challenges around funding and approval and regulatory processes. So for those of you and us working in that public system, what's the best way, do you think, for different teams and departments to join in and collaborate? And I know, Emily, you've been thinking a bit about this in investing in similar public models of innovation and delivery. I think you've spoken a little bit about bringing together kind of consortiums of the different value-adding partners in precinct. Do you have any more thoughts on that?
Yeah, look, it's a really interesting question. I mean, I'm a public servant at heart, very much so, and you know, I've kind of been puzzled at the way that cell therapy has become commercial. Obviously, our biotech and pharma partners have been crucial to kind of push the boundaries of this new therapy, but it just doesn't seem to me like it will end up being the way to go forward. So the way we have done it is to have, really broad conversations through our kind of professional societies to say, okay, how are we all going to do this together? How can we work together? So I'm heavily involved in ICT and also a couple of other professional societies. And these things happen organically. We all talk about it. We say, okay, let's try and work on this thing together. The Collaborative Centres program has been one where we've been working now for 3 or 4 years on how we would do this in Australia, and we've got pilot programs, but as part of those, we've got assessment of health economics. We're engaging with the regulators, the funders. How would we do this in the long term? What's the funding model? And what I would love is if we could work together. I reached out to the whole community of hemoglobin disorders treating doctors, which Michelle, of course, you were involved in that conversation, to say, look, this is in the pipeline. How do we work together in our state and around the country to bring this to our patients? Because everybody has expertise that they can bring to the table. So it's sort of broad collaboration, I guess, in one sentence, Jeanette, is the way I would answer that.
Fantastic. And I'm conscious Richard's appearing to tell me we have one more question. So I'm actually going to use the facilitator's indulgence and ask my own question with a slightly different tact, and that's acknowledging that Sunday is International Women's Day. And to start with, I'd like to express my thanks to both of you for being such incredibly inspiring leaders and world-renowned people in this work for other women in STEM to be aspiring towards. And I've been to a lot of conferences lately and spoken to women who are more junior in their career than I am, who are often getting pretty depressing advice about not pursuing careers in medicine, in science, in research, and acknowledging that it's not all a bed of roses. I thought it might be nice to finish by asking you both what you love most about your job. What do you find the most, acknowledging that there's many tough aspects and the constant kind of slog for funding is difficult. What do you find most enjoyable or rewarding about the work that you do?
You go first, Geraldine.
Oh, okay. Thanks, Emily. It's easy. I love problem solving, and that's basically what my job is. And how lucky am I that doing problem solving also means that I can help people, so that I can help come up with new therapies that will make children have better survival and better health. So easy for me.
Fantastic. And Emily?
For me, I feel immensely fortunate to be able to do what is scientifically so stimulating, and also that it is meaningful. So I love I love my team. I love the environment of the community of cell and gene therapies. So it's an extremely welcoming and rich community of talented people. And it is just so fantastic to be part of that community. And then, the fact that, I mean, Geraldine, you've kind of put it so beautifully, the fact that in addition to that, we're actually doing something meaningful for the community. And I really feel that this type of therapy is going to be incredibly meaningful for families, for children who are affected, for families, adults who have another lease on life because they're able to have treatment. So the tough days then are really enriched by the interactions that I have with my patients. Patients who have been able to go back to work. I have a chap who's gone back out to sea. He works in the Navy, and he had his CAR T cell therapy 4 years ago, and he comes back once a year for a chat, and he's back out to sea completely well, and that just is so rewarding.
What an incredible answer and story to finish on. So I'll hand back to Richard. If that doesn't fill everyone with some hope and optimism for a Friday afternoon, I don't know what will. So thank you.
Absolutely, Michelle. Thank you. And thank you, Michelle, for facilitating the Q&A as well. So we have run out of time. So thank you, everyone, for attending today and for those excellent questions. A video of the webinar will be available via the website and the Sydney Children's Hospital Network YouTube channel. Didn't even know we have got a YouTube channel. There's also a short survey, and Jin's just put up the QR code. So if you could take a moment and fill that out, we appreciate your feedback. Let me take another moment to thank again both Emily and Geraldine for wonderful presentations in what is really cutting edge work, and a really exciting time. It's been a fantastic webinar today. Thank you to Michelle for moderating the Q&A as well. And thank you to the Kids Advanced Therapeutics Program contributors, including the Sydney Children's Hospitals Foundation, the Luminous Alliance, and most importantly, all of you for being here and your active participation. So stay updated for our next webinar by subscribing to the Research Interest Group mailing list using the QR code that Jing's put up there now. And we're done for today. Thanks very much, everybody. See you later.
Great. Thanks,
Thank
Richard.
you.
Thanks, Geraldine. Thanks.
Thank you.